15 students, Essentials of Biology. How are you? It's me again, Karen Klein. Hope y'all are having a nice day. This is a lab simulation. This is about biological molecules. You know, those that make up, or those that are important to living things. We have 4 categories of them. As you recall, we have carbohydrates, which are made up of the monomer monosaccharides, also known as glucose. We have lipids, which are made up of fatty acids. We have nucleic acids, which are made up of nucleotides, and we have proteins that are made up of amino acids. So that's what this lab is reinforcing, that knowledge. As always, please read through this material before you get started, just to give you the background of the information. And the sheet that is uploaded to your lab folder is what you fill out, and then you submit for your lab. So we'll just go ahead and get started. This lab's a little bit longer. And you know, we do this wet lab pretty much exactly how you see it in the lab itself for the on-campus version of the lab. And it takes a while too, but no worries. It's all good. It's all good. Here we are at our lab bench. So let's go through. You have your lab handout up. The first few pages just go through the different categories of those biological molecules, but it also goes through ways we can test for the presence of them. And so I'm going to go through those as well. There's a Benedict's test, and the Benedict's test tests for different types of, or a certain type of sugar called a reducing sugar. And with a Benedict's test, when it is positive, when whatever solution is positive for this reducing sugar, it changes color. Now, Benedict's solution itself is blue. So that blue color, if it remains blue, that would be negative for that reducing sugar. But anything, as you see here on page 2, anything that is something other than blue indicates that you have that reducing sugar. For starches, which is, it's in the same, it doesn't have its own header, which maybe I'll change that on your handout, but there's another carbohydrate called a starch. A starch is a really big molecule. Lots of glucoses strung together, thousands, if not tens of thousands strung together. And there's another test that would indicate the presence of starch. It's called an iodine test, the Lugol's iodine test. And in the presence of starch, this Lugol's iodine, if it's positive for starch, it turns a blue or a black color. Now, if it's negative for starch, it remains the color of the iodine, which is like a pale yellow. So that goes from a pale yellow if it's negative to a bluish black if it's positive. There's a test for lipids. One of those tests, the one you're going to use today, there are a few tests, but the one that you're going to use today is the Sudan test. And Sudan will actually, if there are no lipids present, that the whole solution will just be a uniform kind of pinkish color because Sudan solution is red. But if lipids are present, the Sudan red color kind of binds with those lipids and turns it like a bright red, and that floats to the top of your solution. You'll see when we start. And then proteins is the burette test. I'm on page 3 or 4? I'm on page 3. The burette solution is a kind of a palish color, pale blue, and then in the presence of proteins, it turns more of a lavender color. So for our first experiment on procedures on page 4, through pages, it's actually pages 4 through 6, what you're going to do is you're going to run these tests, the Benedict's, the Benedict's for reducing sugars, the Lugol's iodine for starches, the Sudan test for lipids, and the burette test for proteins. You're going to run it on a positive and a negative control. This is experiment 1. Your negative control is going to show you what a negative result looks like, and a negative control should always be negative. So for your negative control for all 4 of these tests, you're going to run the test on just water, because you know if it turns positive in just plain water, you know that there's something wrong with the reagent. Maybe somebody didn't clean the glassware from the previous class before and there's residual on it. But the main purpose is to be sure that you see what a negative result looks like. And it's the same with a positive control. You run the test on something that will absolutely give you a positive result, so you see what a positive result looks like. And also, for instance, if you're running, the iodine test and you run it on starch solution, and it doesn't turn positive for starch, you know that there's something wrong with your reagent. There must be something wrong with the iodine. There must be something wrong with the starch sample. So we do this one at a time. So I'm on experiment 1 on page 4. And the first one is the Benedict's test for reducing sugars. And this requires a water bath. So we're going to take a water bath from the shelf, and we're going to heat it to 100 degrees. We are going to put 6 mLs of glucose solution in one tube and 6 mLs of water in the other. And we're going to add 6 mLs of Benedict's solution to each of those. And then we're going to heat them up. Generally, it only lets us do one at a time, the hot water bath in these simulated solutions. Now, what you want to do is you're going to record the color of the solution before the Benedict's is added. You see on table 1, we're on page 4. So you're going to record the glucose solution before the Benedict's is added, and then on the next column, after the test is done, after the Benedict's is added and is heated. So we're going to go ahead and do that. Okay, back to the lab. Here's our temperature bath. Set it to 100. Here is our test tube, and this is your positive control. We're going to do your glucose. Yeah. Where's our glucose solution? Okay, there's a burette solution. Glucose. We're adding 6 mLs. Okay, record this color in your table. It's before we add the Benedict's. Let's grab the Benedict's solution. And we're going to add 6 mLs here. Oh, wait, I goofed up. Now you record what the color is, right? Hold on. Record. Before Benedict. No. I was right the first time. Okay. So the Benedict solution is already a bluish color. So just so you know, this doesn't indicate anything yet. We have to heat it. So we're going to go ahead and we're going to heat that up. And in the meantime, I'm going to get, that's your positive control. That's your glucose solution. I'm going to get your water. Going to set up the next tube. Now record this on table 1. And get your Benedict solution. Oh, wait. Benedict solution is right here. Okay. Did I just add 6 mLs of Benedict solution? You know what? I'm just going to add water to that because I just don't want to goof up. Oh, yeah, all right. Okay, so we look here. We're going to take this out. That turned, oh, no, I'm sorry. You put that, record this in your table as far as the color of solution after Benedict's is heated. That was the 5% glucose. Now this is our negative control. This is water with Benedict's solution. And you can see that I wasn't paying attention. So, and we're going to take that out. And you can see there's no, well, you put in your observation. So this is what a negative test looks like. This is what a positive test looks like. But keep in mind with a Benedict solution, as long as it turns any color besides this blue color, it indicates a reducing sugar is present. So if it turned yellow or green or red, that would be positive. All right, let's dump these in the waste. All right, we're going to put them in the sink. There we go. Put the hot water bath. We're going to use you later. Okay, so let's go to the experiment number 2 for starch. Here's table 2. You're going to fill that out. We're going to take 2 test tubes and a 250 milliliter beaker and place them on the workbench. We're going to add 90 milliliters of water and 10% of the iodine. So we're making a solution of iodine because the iodine is pretty dark as it is. So we're making a solution of 90 water, 10 Lugols. And we're going to label that. We're going to take the starch solution and add 6 mLs to one tube, water and add 6 mLs of water to another tube, and record the color of the solution here before the iodine is added, right? Then we are going to add 6 mLs of the Lugols into each, and you're going to record that on table 2 as well, all right? So let's go ahead and get our stuff together. So we're going to get our beaker. I'm going to fill it with 90 mLs of water. All right, get our iodine. I'm going to add 10 mLs of iodine. So we have this iodine solution. We have to label it iodine solution. Iodine solution. Okay, put this over here. Where's our dropper? Oh, okay. All right, let's do this. And we're going to add, where's our starch? No. No. Starch. Okay, we're going to add 6 mLs of starch. I'm going to record your observations in table 2. This is your positive control. Now we're going to, this test tube is going to be water. Oops, got ahead of myself. And record that color in table 2. This is water. Great. Now what we're going to do is, I don't see a dropper up here, so I think it just has you put in, okay. So here's the starch, and we're going to add 6 mLs. Oh, okay. And then 6 mLs. Okay, so here's your starch solution after the iodine is added. Here is your water after the iodine is added. So you want to record those results in table 2. Okay, let's clean up. Hopefully it's going to let us keep that, because we're going to need that later, that starch solution. Hopefully it'll let us do that. All right. Next, we are going to do the test for lipids. We're going to take 2 test tubes from the containers. I'm here on page 5, experiment 3. 2 test tubes. We're going to put corn oil, 6 mLs of corn oil in one, water in the other, and you're going to record those results in table 3. Then we're going to add 6 mLs of the Sudan 3 solution into each of them. Right, so here's our water. That's test tube 2, 6 mLs. And where is our corn oil? Glucose. starch, corn oil. That goes into test tube 1. Okay. So you're going to want to record these. You know, corn oil is kind of yellow. If you do a lot of cooking, corn oil is quite yellow. So record this in table 3. This is before the Sudan 4 is added. Sudan 3. I'm sorry, we use Sudan 4 actually in the lab, which is why I keep referring. Sometimes I refer to Sudan 3. Okay, 4. All right, let's add 6 mLs. Okay, guys. The dog's barking. Sorry. Then we're going to add 6 mLs to the water. Aha! I'm going to lift this up so you see. Remember I said with the Sudan 4 is that it bonds to the lipids. The red dye of the Sudan 4 bonds to lipids and it floats on the top. Do you see that? And this is water. It's just that pinkish color. There aren't any lipids in water to bind with that. All right. That's your lipids test. Make sure you have that all recorded in table 3. Excellent. All right. Are we in there? Cool. As far as our test for proteins, our negative control once again is going to be water. Our positive control is going to be albumin, egg whites, a solution of egg whites, because there's lots of protein in there, and that should give us a positive result. So let's find our albumin. This is the burette test, experiment 4. There we go. Put in our 6 mLs. Gonna get our next test tube and put in 6 mLs of water. There we go. Now record that in table 4. Record their appearance. Now let's grab the burette solution. And we're going to add 6 mLs of burette solution. Now this does not need to be heated. All right, we're going to add. And add. So remember I said that burette solution, this is the water, this is the albumin. Remember I said the burette solution is already like a really pale blue color? The positive result will give you a purple color. And in the wet lab, in the real world, the burette solution doesn't turn that obviously purple. It's such a pale, it's very, very pale and sometimes really hard to interpret. But here in a simulated lab, we're good. So record this in table 4. Now what you want to do is you want to remember these results, because now what we're going to do is we have 2 different food items. We have milk and we have potato juice. Yum, right? And we are going to test both of these for those 4 substances. We are going to test, and now I'm on page 6. Oh, are you okay, Benny? Oh, Benny. I'm sorry. He's okay. Sorry for that interruption. One of the little dogs is blind, and he just fell off the sofa onto a box. He's good. He's still barking. Okay, so you're going to test potato juice and whole milk for the presence of reducing sugars, starch, lipids, and proteins by running each of these tests on both of them. So how we're going to do that is, let's kind of get this all set up. You have a table that you're going to fill out, table 5 on page 7. So we're going to go through each of these tests, and you're going to then want to go ahead and fill out that table based on our results, based on what you saw was the negative result and the positive result for each of them. And I'm going to try not to give you the answer because that's for you to determine. All right, so we're going to go ahead and we're going to run the Benedict's test on the potato and the milk. Let's bring this up to 100. Okay. This is going to be potato, potato, and milk. Okay. All right, let's find our unknowns. Potato juice. So here's our potato juice. Okay. Onion juice, skim milk, and this is whole milk. Whole milk. Okay. All right. And now we're going to add our Benedict solution. All right. All right. So we're going to put the potato in, and we're going to wait about a minute. Will it let us do both at once? It will not. Is it? Nope. Oh, it won't. Okay. Ah, there it is. Okay. Okay. Again, you have to write your results in table 5, or whether it's positive or negative, based on what you know about the Benedict solution and how it determines whether or not there are reducing sugars present. Okay, there's your milk. So here's your potato and here is your milk. So write in whether or not reducing sugars are present, positive or negative. Okay. All right. Next thing we're going to test. Oh, let's put this back. We don't need that anymore. Is we are going to do the starch test. We already have our starch solution here. That's good. So find our potato. Potato juice. Whole milk. Great. All right, and we are going to add 6 mLs of the iodine solution. Remember, we're testing for starch. Remember what was negative, what a negative result looked like versus a positive result? Do you remember? So here's your potato juice, and here is your milk. Okay, potato. Oh, I didn't label them. I'm sorry. This is potato on the left. The milk is on the right. So write in whether it's positive or negative for starch. Get rid of that. Okay. Last but not, oh, hold on, not last but not least. We're not there yet. Okay, let's put that in the sink. And let's put that in the sink. And let's put that in the sink. All right, now what we're testing is we're going to test for the presence of lipids using the Sudan 3. All right, so here is our potato juice and our whole milk. Then we're going to add Sudan 3. Okay. So here's our potato juice. And here is our milk. Now, based on these results, now remember what a positive control looked like and a negative control. Put in table 5 if there are lipids present in the potato juice. This is a potato juice, and this is the whole milk based on these results. All right. Let's clean up. And then last but not least, we're going to check for the presence of proteins using the burette test. Okay. Let's get our potato juice. Actually, you know what? We can put right here with the burette solution. Is it gonna just? So I'm gonna put the burette in first. See if it's gonna. I'm just curious if the computer is gonna give us a problem. That would be really weird if it did, because obviously, I mean, potato juice. Okay, potato juice. Okay, so here we are. Let's add the potato juice and the whole milk. All right. So this is the burette test, testing for the presence of proteins in potato juice and for milk. So you want to write positive or negative, knowing what you know about the burette test, in table 5. Okay, well that's it for this lab. Thank you for participating. Now remember to check over your sheet, make sure everything is filled out, the things that need to be. And once you're comfortable with your answers, you will want to add that to upload it to the submission link, and you're good to go. Take care, and we'll see you later. Bye.